Yesterday we have seen the 5' methyl guanosine cap. Today we will study the 3' polyadenylation in eukaryotic nascent transcript RNA. And then the next day we will talk about splicing. MSc. Okay, so let us talk about polyadenylation. So this is, you can see the DNA strand over here. On the top of this DNA strand, you have the RNA polymerase, which is synthesizing this messenger RNA. It has got a 5' cap already attached. And then gradually you see a particular sequence coming out, AAUAA. So this is supposed to be a polyadenylation sequence. And immediately after this sequence, kachak, there is a cleavage over here. And then this mRNA is stabilized by polyadenylation. So at the end of the mRNA, a series of A is added. Because there are lots of 3' exonucleases which can chew through these mRNA. Before the mRNA does a process of translation and makes proteins, it can be chewed up by the nuclease or the DNases which are present in the cell cytoplasm. Essentially, they were to destroy viral DNA which comes inside. So for that, but these protect this polyadenylated tail. They protect the mRNA from degradation. বুঝতে পারলে? হ্যাঁ না?
Yes, sir.
Yes, sir. Yeah, yes, sir. So the 3' ends of polymerase 2 transcribed mRNAs are generated by cleavage followed by polyadenylation. So যেই AA সিগন্যালটা আসে, হ্যাঁ, যেই ওটা ট্রান্সক্রাইবার খাচাক করে কেটে দেয়, কাটার পরেই পেছনে A অ্যাড করে দেয়. So this is how it works. The same thing. So the hexanucleotide sequence AAUAA is necessary for cleavage to generate a 3' end for polyadenylation. So a sequence, the moment this particular sequence comes, an endonuclease comes and kachak does a cleavage over here. And then the mRNA is stabilized by polyadenylation. এর পেছনে A আবার অ্যাড হয়ে যায়. The unprotected 5' end gives signal for transcription termination. এইখানে দেখো অনেকগুলো A অ্যাড হয়ে গেছে. A to the power n হয়ে গেছে. So there is no poly DT sequence on the DNA template. Normally DNA template থাকে না. So this polyadenylation or the polytailing, it does not depend on the template. And this tailing process, it occurs before splicing happens. So I am talking about, I'm taking classes in the sequence as it happens. First, 5' methyl cap happens. Next, polyadenylation happens. After this, what will happen is splicing. And the site of polyadenylation is in the nucleus. There is this enzyme PTEF-B2 kinase. It phosphorylases S2 at CTD tail, the serine 2, at the C terminus of polymerase 2. And this recruits the machinery required for 3' polyadenylation and splicing. Termination of transcription occurs only after RNA polymerase has transcribed past a consensus AAU-AAA sequence, the poly A addition site. 10 to 30 nucleotides pass this size. A string of 100 to 200 adenine residues are added to the mRNA transcript. The enzyme is poly A polymerase. They add these adenine residues. From Garrett and Grisham, this picture. RNA ends AAUAA after 10 to 30 nucleotide downstream. Gachak, there is this cleavage, and poly A polymerase, it adds the next. So this is the DNA from which the RNA polymerase binds, it goes, it ends. Here the messenger RNA is produced, the cap is added. After this AAUAA sequence, 10 to 30 nucleotides after that, there is a Gachak cleavage taking place, and this particular nascent RNA is produced. And after that, poly A polymerase adds around 100 to 200 adenine residues after this. বুঝতে পেরেছো?
Yes, sir.
Okay. The same thing, this is the cleavage signal, polyadenylated precursor. So there are two components. One is the cleavage component, the other is the polyadenylation component. So this is the polyadenylation recognition sequence in yeast. Before that, there is an efficiency element present. In mammals, there is an auxiliary upstream element, then a variable length, then a polyadenylation sequence length happens. Then 10 to 30 nucleotide after that, there is this poly A site. Maybe there is a U-rich region over here. So, this is polyadenylation. In the next class, I will talk about splicing. Do you people have got any questions regarding polyadenylation? I believe you have the note. কোনো প্রশ্ন আছে?
না স্যার।
ঠিক আছে, তাহলে আজকে বাই বাই।
ওকে স্যার, থ্যাংক ইউ স্যার।
Thank you, sir.
Welcome.